See, if we don't have coffee, we can't go on, right? So everybody gets tired anyway. So, yeah. Originally, Kristen asked me to give a talk on some of the latest things in genomics. But I think you probably have enough talks by now, and enough talks about, especially the great talk by Danji. So I thought I'd just go over some things that I heard discussed during the last 2 days of people asking questions about genomics, and so maybe we can just discuss this. And I should say that I made 5 questions, and I'm not an expert in any one of them. And so I'm glad Dan's here. Maybe Dan can answer some of these questions as well. You know, biology is really complicated. You have to learn so many facts. My son recently took the biology SAT-2, and of course I had to study with it. And I think he did a bit better than me. So, you know, there's so many different things you have to know. All right, actually, so Dan's talk was really a great introduction to this question, which I think came up several times, which is, in fact, can genomics predict disease risk? And I think Scott talked about how 23andMe had this idea that they could do that and sell you that information, and then it kind of got shut down because the FDA thought that you can't really do that with sufficient precision to be able to give you information. Okay? In fact, 23andMe actually has a very interesting follow-up to this, because a lot of times what we can and cannot do by knowing your genome really is limited by the amount of data that we have, and large part of the limitation in the data is so-called phenotype data. Okay? So genomic data we can get fairly easily now, and especially for important loci, we can get them for a few $100 instead of a few thousand, which is what 23andMe uses. What's harder data to get is everything about you besides your genome, right? And because that's much more amorphous, how do I measure it? Do I take pictures and study it? How do I know when somebody has a disease or don't have a disease? So 23andMe right now, they collected about 500,000 genomic information from people, mostly by selling the idea now. They have the new model is that they'll tell you something about your genetic ancestry and composition of who your ancestors were and things like that. Actually, I guess I had no idea that this was such a popular idea. Even my wife sent away for the kid and did this. But then they did this interesting experiment that they have 500,000 genome data now, and they have reserved the right to contact you with questionnaires. They intended, so they done contacted people and asked, do you like cilantro? And as you know, some people, when they taste cilantro, it tastes like soap to them. This turns out to be a genetic trait. And by doing this and asking the people that they already had on the database, they immediately identified the loci which was responsible for cilantro. And you can imagine that they could go on to do this by, did you have cold last week or something like that, right? Like they asked this question of susceptibility to particular diseases. I guess, large part of this, the problem is, what problem that Dan discussed. We have 2 problems in what can you tell from the genetics. One, as you already know, is the so-called heritability of any given trait. That the trait is comprised not only of genetics, but also the environmental factors. So sometimes we also call this penetrance of a trait. And heritability of traits varies widely, even in disease-related traits. So like autism, and most of these heritability is in humans, especially where we can't go and make humans at will. In animal systems, we can make the genotypes that we want and get a very good estimate of heritability. It's harder to do that, of course, with humans. A lot of these studies are done, the estimates come from studying twins. So monozygote twin concordance rate for autism is up to 90%. It's one of the more heritable diseases. Depending on what you mean by autism, of course, as well as something like schizophrenia, something like 20% or so. So there's that variability. The other part, as also Dan said, is the number of genes that might be responsible for that trait. And so the monogenic trait, or what we standardly call Mendelian trait, and these are most of the things that come out in textbooks. There's right now, for disease-related traits, there's a few hundred diseases that's supposed to be monogenic, and there's a database for this. Those are the easiest, of course, to try to predict from, and those are the most important traits that when you go in, as they have genetic counseling for a disease context, then they will be able to tell you something about things that are monogenic traits. Unfortunately, they typically also tend to be rare. And not many people are affected by these kind of things because they are Mendelian and because they're single locus and they have typically high penetrance. It's that those disease allele variants are just not prevalent in the human population due to evolutionary pressure. Okay? So what is hard is these complex traits which have many loci as the underlying cause. Autism. Initially, people thought it might be few loci that have autism. Now the estimates are that it's up to a few hundred loci that are potentially underlying autism. So this is why it's very difficult to predict disease risk. And that's what we would all like to do. And most of this problems is really data bound. So I think it's no doubt that as people do more and more clinical sequencing, that part will really improve going into the future.
I think regarding 23andMe, also like recently I read there, now they could also sell your data, so now companies are going to them for all your data and for the same idea, I guess.
Right, that's right. And in fact, those they're charging like $99 or something like that right now. They actually cost about $230. And so they're taking a loss at every kit that they sell because their business model is to sell your data.
So does that make it a good deal or a bad deal?
Now that comes to the legal, ethical questions about genomics, which we can discuss in a moment. I don't know. Selling information, that's a very hard business model, by the way. And many genomics companies have started out with the idea of selling data. And people don't want to buy data. So the originally the company that Craig Becker set up, which was one of the drivers of the Human Genome Project, and it's because of the kind of technologies that they set up that really accelerated the completion of the Human Genome Project. And their major idea was that they were going to sell the information to companies and such, and that didn't really work out in some business model. So, I've also had questions before from lawyers about, well, can you use genomics to do paternity identification and sibling identification? That certainly is true. That degree of identification is quite easy and pretty standard as well as you know. Ancestry is more complicated because we're all very mixed population, even though we may be phenotypically relatively distinct. People question the idea whether there are such things as races and so on. There's no doubt that there's clusters of phenotypic traits that go together. However, if you trace, people have done rough model-based calculations, and if you trace back just about 25 generations or so, every single individual human shares some part of the loci as the ancestor about 25 generations ago. Right? So Marie and we probably come from very geographically distinct ancestors, right? But we definitely share at least some part of our genome, with high certainty 25 generations. And that's not very hard to see, right? The reason is that I have two parents, right? I have 4 grandparents, and then I have 8 great-great-parents. That number of ancestors goes up geometrically as you go up into the path. So by the time you go 25 generations, what is that? That's 2 to the 25th power. So that's about 32 million ancestors that I have, right? Each one of you have 32 million ancestors. Somebody has to overlap somewhere, right?
So what you're saying, sooner or later the paths. So what you're saying, sooner or later the paths cross.
Exactly, right.
They have to.
They have to. So there are people who think that you can predict phenotype from the genomic data, and this comes out in like forensics, right? So you can collect DNA from a crime scene, and can you reconstruct like the person's facial morphology and phenotypic traits? So, I don't know, Dan, what do you think about that possibility?
Probably not too well. It's, again, it's interaction with your environment and your developmental history.
You can probably tell whether you're Asian or Caucasian or something like that. Things like eye color genetics is reasonably well done. Other traits, I'm not sure.
Sometimes in college newspapers on Ivy League campuses, they advertise for females to get their eggs. So
Yeah.
are they pretty confident that they can get a particular phenotype?
Boy, that's a very loaded question, right? So the whole active.
And I think it doesn't really matter because what they want to buy is the Ivy League pedigree.
Right.
Because the people buying the eggs probably have little to no understanding of genetics. They think
You
they
think
can.
that they want to be able to say, I don't know who the mother of my child is, but we know there was an Ivy League.
And let me tell you, once the child is born, you realize it's the smartest child that was ever born, right? So it works.
You know, actually, I'll go over the genetics of so-called desirable traits, right? Like IQ or whatever it is, or athletic abilities. Those are all such taboo subjects for people to study. That there's very, very little research that actually goes on, and very, very hard to say anything. Yes, so if you come to this, can genomics predict disease risk or phenotypes and so on? Well, then of course there's this question of can genomics help with therapeutic strategy? And I don't think I need to go over this because Dan really just went over a large parts of this. The new drug development question that Dan asked, what is surprising is that although there's hundreds and hundreds of drugs out there, the actual molecules in your human body, your body, that the drugs target is, I think something like last time I said, it was like a dozen. Some are like
Within an order of magnitude.
yeah, it's under 2 dozen for sure. So hundreds of different drugs, but the molecules that are targeting your body is about 2 dozen or so. So this is so the idea that somehow all these drugs we need to find new target. This has been a very, very difficult problem for all the different reasons that Dan talked about. Accessibility of the target, the toxicity of using compounds that do the, hit the target. What is different now is all the biological drugs. So it's like protein-based drugs, by which we mean things like antibodies, or now things like coming out like RNA-based drugs and so on. Those, of course, have much more wider range of targeting. But the small molecule drugs, a very small number of targets right now. And that was originally the idea, holy grail of genomics was that if we understand all these different pathways for diseases, maybe we can find all these different drug targets. And that hasn't really come out to be, up to now anyway, been realized.
I have a question, and perhaps it would be for either you or Dan, but when he was talking about the drug pipeline, I began to wonder about the Zika virus. First, I want to know if I'm correct in understanding that the Zika virus has been around for a while, and it actually started in Egypt. Can you confirm that?
I can't say off the top of my head. I know that it's been around for a while in the sense that, you know, it hasn't cropped up in the last 2 years. My understanding is that the recent explosion in Brazil has been sort of a strain that has evolved from a different continent, and it only arrived in Brazil within the last decade or two.
Okay, so with that being said, how long do you think it would be before a drug to combat the Zika virus is available on a large scale?
So if you look at the chart, it would be on average 10 to 15 years. Let's hope it's faster than that. I know that there are several vaccines companies that are trying, you know, that have dedicated resources to Zika in particular. But
so is it possible to with the Zika virus? Let's pretend for a moment that the goal was to get the Zika virus under control before the Olympics, not in 3 weeks, but let's say the Zika outbreak happened 2 years ago, and they said, well, we have the Olympics in 2016, it's 2014. Is it possible to be able to fast track the drug?
so there are fast tracks for certain drugs, and it's often the case in you know, diseases of highly unmet need or late-stage terminal diseases like cancer. You can, you know, the problem is if you have to go through a clinical trial, then that trial, recruiting, actually doing the trial and analyzing the results and then submitting the results to the FDA for approval, that in of itself really can't be done faster than 2 years. So unless there's an avenue that a government provides that's a faster track than that.
Like they did with the AIDS.
So,
Certain situations, the FDA may make some exceptions. Like with cancer, if you find even the most minute improvement, they're going to approve it. They're going to approve, FDA usually will approve because the widespread of how many people, there's, what is there, 1 in 2 chance of the people in this room getting cancer? So anything in cancer is usually a fast track to some, but like you said, to only some degree, unless you, the drug company, can justify, or the mass at large, like with the AIDS, they were able to fast track a lot of the drugs, and some of them didn't pan out exactly, if I remember, because they were too fast.
Didn't that happen with Ebola? Wasn't there an American company that treated the 2 Americans that were brought back?
yeah, so I'm not terribly familiar with the backstory for Ebola, but yeah, but I think because of the past outbreaks of Ebola, there was sort of a whole history of research into the virus.
The antiviral therapies are different from the vaccine, so it's not like a small molecule with a whole lot of unknown toxins the biological reasons. And Zika virus, there are right now fast-track trials going on with vaccine for Zika virus.
Isn't one of the use for genomics in looking at whether or not certain groups of people are going to be more, certain drugs will be efficacious more in X group than Y group?
That's right. So as Dan said, you know, of course, if you do a drug trial, you can find that certain groups of people more, the drug is more efficacious or has less toxic side effect. In fact, now, so these are called companion diagnostics. That is that now there's about 100-some drugs now, where the FDA has approved the drug with there is a genetic-based companion diagnostic that this drug is most appropriate for this genetically group. For example, cytotoxicity depends on the drug metabolism, which can depend on things like the status of the cytochrome P450 gene. And so there are the high metabolizers and low metabolizers. So that is in fact part of this targeted therapy idea. But the more important targeted therapy probably is, and the most applied right now is cancer. as we already discussed. Every tumor is different, and in fact, in a given tumor, there can be many heterogeneous clones in the tumor. And so if you have cancer, right now, the genomic screening has just been approved for medical reimbursement. That just happened like beginning of this year. Prior to that, the doctor can order what's called laboratory developed tests. So meaning that they can custom order a screening service, which Penn provides. There's some companies that does that, but that's typically not insurance reimbursable. Then the doctor has to go through this long rationalization as to why they should do it. However, if any of my family members had cancer, or if I had cancer, that would be the first thing I do. Because although the hit rate still is fairly low, so the hit rate, so if you do a genome-based screening, and the whole reason to do that is to try to see what driver mutations might happen in your particular tumor, and whether there are specific drugs that can inhibit those driver mutation pathways. And that may be very different from your standard therapeutic regime. The hit rate right now is somewhere between 10 to 30%. So that means that if they do the genomic screening, they can find the driver mutation that they know about. Even though you do the entire genome, FDA does not allow you to make the diagnostic on the entire genome, so there's some specific loci in which you can make the clinical diagnosis. And then there has to be some kind of drug that's available that can target that. Sometimes they do actually the screening de novo, that is because there may be 100 compounds that are FDA-approved compounds, but not known to affect a particular target they
Well,
think
then you
that they
take
have.
the guessing game out for the doctor, is what you're saying.
Well,
To
guessing
some
they-
degree.
Well, to some degree, that's right. And so they can do
That's what
actually,
you meant by the 20 to 30%.
right. They can do the screening de novo as well. And you can get, 30% of the time, optimistically, you can get a completely new drug treatment that the doctor would not have sought to do before. And of course, 30% can be miraculous for a certain number of people.
Is this something that like any oncologist would be able to do? You would just ask the doctor could they do it, or would you have to be part of a special health system? Yeah.
This gets to a very tricky area, which of course, I guess there's no doctors here. Not every doctor would do it, right? And sometimes it's, also there's the reimbursement problem as well. So that's another factor. The reality is that the U.S. medical system is, the system is set up in such a way that the doctor is the king or the queen, right? So that's why, for example, the doctor can prescribe off-label uses for drugs, or they can, or every surgical procedure that the doctor chooses to do is their own autonomous decision as to go this way or that way. So in some way, in any of these new therapeutic regimes or the drugs that's developed and so on, what really needs, there's no association that says that if you have cancer with KRAS mutation and triple negative or something, you should use this drug. This is doctor's choice.
How you put that, because the doctors I've talked to, they say it's the insurance that weighs heavily on their decision.
That also happens, of course. The doctor is the point of the decision-making contact where they have to balance balance what's the standard therapeutic regime, what might be known as the latest thing in the literature, what's reimbursable or not. And so then when you talk about something like genomics-based therapeutic screening for, say, tumor, some doctors buy into it, other doctors don't buy into it. They think that it's unnecessary, right? And so so a lot of the medical practice in this country is not systematized in some kind of this is the best thing to do.
Are they actually biopsying the tumor, or they just test your genome?
No, they're biopsying the tumor.
Okay.
And also those companies follow through on the therapies, so their databases are growing, as they're being used too.
That is true.
one other question. Do you play devil's advocate? Do you think it'd be a good idea for healthcare, what Dan was talking about with the drug basis, that we have a national genetic database? Would that help in drug design?
There already are a lot of such databases.
Right. But I'm saying one that would be national. In other words, every child born down the
So
road.
certain countries already have that. So
Yeah, exactly.
Finland is a really good example. They have
And-
a very extensive national health database that's now including genetic information.
Now,
that-
has that helped?
That they're working with U.S.-based academic institutions to use to look for, the genetic basis of different traits.
On how that helps them in, let's say, drug design, for example.
Right, and another good example of that is the Icelandic population. Which I think was the total population size was like 200,000 or something like that, the entire population. And they're very genetically isolated, founded by Vikings who ended up there. And they have an extensive pedigree, and they have sequenced pretty much the entire population and then formed a company based on this information. And that has been for the past 15 years or so the go-to place to try to dissect the genetic basis of diseases. Which kind of gets to this issue over here, right? So, can the genomic information used to invade my privacy? Right? And especially things like, what if you can't predict disease risk, like that? Can private companies use that to do things like, deny you insurance coverage, deny you admissions to employment, absolutely. Right? In fact,
Or we're only employing to, okay, we know that because of that gene or something like that, you're going to get a specific cancer at a certain age, so we'll, eliminate your job the year before or something. Don't laugh, these
yeah,
questions come up.
so currently there are laws in place. There's something called is it called GINI or
Genome.
Yeah.
Right? So genetic information, which is supposed to govern the exchange, the limit the exchange of using such genetic genomic information for those kind of purposes. I don't know if that actually strictly that people don't do that. But it's certainly clear that genomic information can be used to assess risk, right? And for that, and for good in the sense that assessing your personal risk, but certainly somebody else can assess risk of you.
Well, they could use it as a weapon for service.
There's when we do genome-based studies with humans, which we do right now, as I mentioned before, there's a lot of institutional review board processes that we have to go through. Then we take that information and we deposit it into national databases, as Jim said. One of the issues that come up is that these kind of data, when we do deposit them, anonymized, so that there's no personally identifying information that's attached to the data that we deposit, so that they can't be traced back to you, and somebody goes and analyzes this expression data we deposited and says, this person, it turns out to be Jim, has this particular trait or something like that. And so in principle, you're not supposed to be able to do that. In practice, of course, your genome has the fingerprint of who you are. Right? So you can look at these databases, look at the specific genetic variation that's in the databases, and those variants can become unique identifiers. Okay? And if that information exists somewhere else, you can cross-match and eventually get back to finding out the identity of the person. And that is a very definite possibility.
Well, what about when some criminals are apprehended, if they started off in Florida and they end up in Pennsylvania? Isn't there a national database where they match the DNA?
There is there's a set of more technique called DNA fingerprinting, and that is a particular set of markers in the genome which is supposed to be unique to an individual, and since the data sets exist with DNA fingerprint stuff, I don't know about overall whole genome information. I don't know of any tracking of whole genome information in the criminal justice system, but then I wouldn't know. Which sort of gets to things like, can you sort of track skin cells that give off and all everything we give off, all the biological materials that we in our body has genomic information in it. That's it. So, and we can recover genomic sequences from few skin cells. That is definitely technically possible. So it's not impossible. It might be costly, but it's not impossible for all of us to leave this room and somebody to go around, sweep the floors, and then figure out who's been in here. That's a technical possibility. And this is sort of the more and more controversial question, and the question of human engineering. And as you know, people do. There have been many attempts to do genetic correction, genetic defects, and Dan talked about gene-based therapy. There have been other kinds of, actually recently I guess there was approval for somebody with mitochondrial disease. I don't know if you saw that in the news. But the mitochondria is a maternal origin. So they approved of therapeutics where they will take a third person's mitochondria and use it to replace all the mother's mitochondria as a therapeutic treatment. If you can do gene therapy, which we can, and we can do it at least much better than we used to be able to do. The hard part of all of that is really, you can do all kinds of gene therapy at the embryo level. So the hard part of the gene therapy is really at the adult level, being able to deliver the corrected gene into all the billions of cells that might need it, that might be an organ that might need it. Correction. And all the problems previous to that came from not so much in the gene therapy itself, but the delivery system used to do the gene therapy. Right? So the only way we can deliver to a billion cells is to do something like use viral vectors, so have the viruses infect those cells and deliver the load into it, and that can have all kinds of negative consequences. But if you do embryo engineering, which can be done, then that's technically definitely feasible to do. And I think Sean mentioned something like CRISPR technology. So CRISPR technology is the technology. It's not that you couldn't do it before. One of the technical problems was doing this kind of genetic correction or changing the genome is that let's say that I have some gene that I want to change it to mutate to a different gene that used to be brown eye color, but maybe tends to give you blue eye color, something like that. You could take that piece of the gene and put it into the genome. It's very hard to place it exactly where replace the original gene. then they go into some different part of the genome and create some side effects that we don't really know. So ideally, if you want to sort of correct the gene, change the genome, you want to change it exactly where you want to change it, and in exactly the sequence that where you want to do. So that technique challenge has been there. In some organisms, that's easy to do. In other organisms, it's very hard to do. Some organisms, it's almost impossible to do. And the reason that everybody's excited about this CRISPR technology is that this technology seems to work in pretty much all the organisms that we work with, and therefore we are now able to, even with human genomes, be able to say, that particular base pair out of this 3 billion base pair, I want to change it from an H to a C, and that doesn't require a whole lot of trial and error that we used to do. So we can do it.
If I were doing it, I'd still have a good lawyer.
Pardon?
If I were doing it, I would still have a good lawyer. How would you take care of the liability if we screwed up?
Oh, yeah, well, I mean, of course, and you're way beyond liability questions. We're really talking about ethical and moral, and of course, legal questions. Right?
Something that I wanted to say, so back when the eugenics movement was happening in like 1920s, 1930s, the Supreme Court said that it was absolutely legal to remove someone's ability to reproduce. And then so that moving into the future, if we are finding genetic defects in people, that Supreme Court ruling was never taken away. That Supreme Court ruling is still it's still active. So what's to say the government can't just decide that because you have these specific flags and these specific markers that you are not suitable to reproduce, they technically legally still have the authority to do that.
Although they have it right now, and they're not using it.
Yeah, they're not using it now, but they did in the past. And like up until, like I was saying before, like up until like the 1970s, they were still actively doing that in Puerto Rico. So I can see how that could easily become an issue. Not that it's definitely going to be used. I think the main reason why they haven't retracted it is because they don't want to get sued by people that are still alive that were affected by it.
Yeah, of course, the legal and moral issues with respect to these kind of things is a great thing to discuss with students. At the moment, I just want to present to you that the technical barriers, none of this is technically possible. The question is whether one should do it or is anybody doing it. For example, one of the things that's happening now is for in vitro fertilization. As you know, a large problem with in vitro fertilization is that you do the in vitro and then you don't know which egg is going to develop properly, so that people get implanted with say 4 or 5 or 6 or 7 eggs, and then that's why you end up with quadruplets and so on, because all of them actually have successfully implanted. As well, you don't know whether any of them might inherit some the parental defect. So what's become one technology that a lot of people are working on, and whether this is again morally right thing to do or not, is so-called pre-implantation testing. Right? And in fact, the IVF doctors already do pre-implantation testing. They have tests sort of that tells them which one of the fertilized eggs look like the most potentially viable and so on. Well, now you can do pre-implantation genetic testing. What they do is that as the fertilized egg grows in the dish, and once they get to about 8 cells, they pluck off one cell. They claim that that's okay. I don't know whether that's actually okay or not. And then they can do genomic testing on that single embryo. Right? And so that leaves open all kinds of things like maybe you're not going to do genetic engineering, but you can avoid all of those variants that you think you don't want. And, so those are all certainly technically possible and rapidly developing right now. are on time.
We're running about 15
Oh.
minutes before that, yeah, so.
Well, all right, I'll just put this out there. Okay. So, yeah, maybe we don't have time. So this has definitely become an issue as well. Because, for example, one of the issues that came out about 3 years ago is looking at bird flu viruses. People started to, because the bird flu typically don't transmit to humans, and when they do, that's when we have flu epidemics oftentimes. So then people study what are the factors that allow the bird flu to jump to human, and they actually found like a couple mutations that allow them to do it. And the question, this was a very important question, is whether you should allow such information to become public or not. Because maybe then you can go and engineer other viruses to jump into humans.
I think MRSA is considered a superbug.
Right. That's what, MRSA, you mean the multiple resistance bug, yes. Well, you can certainly do that too. Engineering those kind of bugs are not that hard to do. I think recently somebody asked me, as these things like CRISPR technology came on, can we now just genetically target specifics of populations of people for bio warfare? I don't think that's possible. So you can certainly engineer, lethal viruses or lethal microorganisms, but genetically targeting such things is that's still science fiction.
But they say what science fiction now soon will be a reality.
Hopefully that's.
And earlier today, I heard Gattaca referenced, and when I'm introducing genetics, I show Gattaca, so I will tell you that I've probably seen Gattaca about 50 times, like the whole film. And there's one part where Blair Underwood, when the parents are pre-selecting the traits that they want, and then the mother says, well, we want to leave some things to chance, and he says to her, why would you want to do that?
Yeah.
Still, just the best of things.
I'm sorry?
I've seen it about 40, 50 times.
But a large part of the things that are in Gattaca certainly is possible. It's technically feasible.